Hi everyone and welcome in my channel. In this tutorial video, we will talk about antigen-antibody interaction in vitro. So we will talk about heme agglutination, which is agglutinate of red blood cells as a result of antibody bindings. We will talk about the two stages of heme agglutination, which is a sensitization step. Also talk about the other step, which is lattice formation, and we will discuss factors affecting these two stages and their importance and how we can manipulate these factors to affect the stages. So let's start. So heme agglutination, as we said, happen in two stages. The first one is the sensitization step, and the second one is the lattice formation step or the agglutination step. So in sensitization step, the antibody will bind to the corresponding antigen if it's present, and this is a reversible step. Then bridges will form between red blood cells, causing the visible agglutination. So, we can see the sensitization step by our eyes. However, we can see the agglutination steps. So this is the end stage of tests. So if there is no RBC antibodies in the serum, or if there are no corresponding antigen to the exist antibody, no agglutination will happen. So in sensitization step, the antibody will bind to the corresponding antigen on the red blood cell surface. Let's have a closer look. This is the red blood cells, and here the molecule, the antigen on the surface of the blood cells. The antibody binding site will binding to a specific part on the antigen molecule. We call it epitope. To illustrate this point, let's talk about Duffy as an example. Duffy molecule spins the red blood cell membrane several times, as you see here. And here is the epitope or the antigen determinant site on the Duffy molecule. So if the antibody anti-Duffy A, for example, will bind, it will bind to this particular part. We just explained the sensitization process, and now we will talk about factors affecting this stage. We will talk about temperature, the incubation time, pH of the environment, and finally we will talk about ionic strength and how it affects. Let's talk now about temperature. There are two types of antibody involved in hemagglutination process, which is IgM and IgG. Each one of them has optimal temperature to work at. For example, IgM work better at cold temperature, so it's work at room temperature or lower temperature, while IgG need a quite high temperature, so it work better at 37, which is the body temperature. So we don't expect IgG to work at room temperature, so it will not bind to the corresponding antigen without provide the optimal temperature. Let us now move to the other factor, the incubation time. Some tests and reagents require a specific incubation time to allow for antibody to bind to the antigen. However, other procedure may require immediate spin, so there are no incubation time required. pH also play an important role in the sensitization step. The optimal pH for antibody to bind to the corresponding antigen is the physiological pH 7. So, by manipulating pH, we can dissociate the antibody from the antigen. So, if we add acid to reduce the pH, for example, and reduced from 7 to 6, the antibody will dissociate from the corresponding antigen. The other factor is the ionic strength. In an isotonic environment, such as the physiological saline, sodium and chloride ions are attracted to the opposite charged group on antigen and antibody molecule. So sodium, because it's positive charge, will attract to the negative charged molecule, while chloride ions are negative, so it will attract to positive charge. As a result of this attraction, the combination of antigen and antibody is hindered. If the ionic environment is reduced, so there are a lower amount of ions, this shielding effect is reduced and the amount of antibody uptake onto the red cells is increased. So we will talk later how to use low ionic strength saline to enhance the agglutination and its work by this principle. We just cover the most important factors affecting the first stage and the antibody binding stage. Now we will move to talk about factors affecting lattice formation stage. So we will talk about the distance between red blood cells and zeta potential. We will talk about optimal concentration of antigen and antibody, and finally talk about effect of centrifugation. Red blood cells at a physiological saline carry the negative charge as a result of presence of abundant amount of sialic acid on the surface of the red blood cells. So as a result of these, the red blood cell will repulse each other. So there will be distance between each two red blood cells, and this distance is corresponding to the zeta potential. So for this is reason, IgG and IgM have different ability in agglutinate red cell at a physiological saline. So because IgM, the binding site, the distance between two binding sites is quite large, so it can cross the distance between two red blood cells and cause direct agglutination. However, IgG is quite small, so it can't cross the distance, so it can't cause direct agglutination. So they need enhancement media, which we will talk about it later, to cause agglutination. In order to reach the lattice formation stage, the antibody and antigen should be at optimal concentration. So there are no excess antibody or no excess antigen in the test. So if there are too many antibody, the antibody binding site will be saturated, which leading to false negative result. So to see the agglutination, we have to optimize the ratio of serum to cells. And in practical, we generally can achieve this by adding 2 drops of serum to 1 drop of 5% cell suspension. We prepare the cell suspension because 1 drop of blood contains very high percentage of antigens, which will affect the optimal concentration ratio, leading to missed the reaction. So we will have again a false negative result. Centrifuge effect is the last factor I will talk about influence the second stage of agglutination. So when we spin blood tube in the centrifuge, the red blood cells will come close to each other, which will enhance agglutination as we discussed before. But however, sometimes when we take the blood tube from the centrifuge, we can see a bottom like. So people without experience may consider this as +4 agglutination, but in fact, they have to gently flick the tube because this is sometimes the effect of centrifuge. So the cells may suspend again in the tube, and maybe it's true for positive result. So people without experience have to be careful when they record the result. In this video, we talk about heme agglutination and how it's happened and what's the stage involved in heme agglutination. Also, we talk about factors affecting on the antibody binding step. We talk about temperature, incubation time, pH, and we said by changing the pH, we can dissociate the antibody from the antigen. Also, we talk about ionic strength and how it affects. Then we move to factors affecting lattice formation. We talk about the distance between red blood cells and the effect of this distance on agglutination and how IgM perform differently than the IgG. And we talk about optimal concentration and their importance in detecting the reaction. And finally, we talk about centrifuge and how sometimes we read the result in a wrong way. So thank you to watch this video and see you in the next video. Thank you.